Good evening, ma'am.
Good evening, sir.
Yes. Thank you, ma'am. Thank you so much. Good evening, my dear students. Once again, you are all are welcome to the biological session. Last class, what are the things already we discussed? Embryo culture, meristematic culture, and also somatic embryo regenesis. Otherwise called as regeneration. Idea. So, today what we are going to discuss about, remaining of the culture. What are the culture? So, anther culture. What is the meaning of anther? Already you know very about anther is nothing but the male reproductive part of the plant, isn't it, my dear? A flower, isn't it, my dear? So the anther, how it will be grow in the laboratory condition, that things we are going to discuss about. Not only that, and also which plant they were taken for an example or sample, who is first introduced in India. So all those things we are going to discuss in this part. First of all, you know about what is meant by anther. First, you should note it down. So what is meant by anther? Take any part of the plant. Take any part of the plant body. It contains, there is a center. What is that, So one of the ovary, isn't it? Completely collectively called as ovary. This is consists a stigma, style, ovary. Isn't it, my dear? So ovary. So followed by what happened? There are two, what is that? Anther present in it. Depends upon their, it may be vary. According to the plant, it can be vary. Okay. This one is called as what is that? Female part. Female reproductive part. Isn't it? This one is called as anther. So otherwise called as male reproductive part. Followed by which one it has already know about that. There are, what is that? Taken examples. Corolla is there, and followed by there is a green colored, the outermost layer is called as what is that? What is that? Calyx. Calyx, corolla, andrecium, and gynoecium. So here the anther is also very, very important. So already in our little anther, it contains the pollen grains, isn't it, my dear? Pollen grains. When the pollen grains undergoes the decussation process, already know the anther, when it undergoes the decussation process, it emits a lot of pollen grains. When it reaches the stigma, automatically the pollination process occurs. Pollination start. After that, you know about what are the physiological changes which occurs within the flower. Already you know about the fallopian tubes. Already you know about there is a nucleus also move on to the male gamete also move on to the until the reach of female gametes. Already you studied the triple fusion, isn't it, my dear? That things also already we discussed. Okay. Here, the anther is also culture in the laboratory. See that the male part is also under laboratory concern. We can produce that things we are going to discuss in deep manner. Okay, see that one. So that the haploid are produced in many plants species when their anthers are cultured on a suitable medium. So this is also very, very important, my dear students. This is also a very, very important part. So anther already in our it is also nothing but the haploid nature. It's only N condition is there. And male reproductive part. And female also have it has the, what is that? Same haploid nature is there. Both are come in contact close together During the, what is that? Fertilization process, what happened, the N plus N, it gives 2N. That is called as diploid conditions, already you know what. Okay. So the male is also denoted as a symbol like this, haploid nature. And female is also this one. Okay. This one is called as male. This one is called as female. Okay. The anther is the microspore. Okay. Otherwise called as pollen grain. It has a N, and also it also has N stage. Both are common contact to you will get diploid zygote, isn't it? Here, the anther, it has a haploid nature and produce many plant species. All the plants, all higher plants, particularly the angiosperm, it contains N nature only, haploid nature only. When their anthers are cultured on the suitable medium, this is called as anther culture. When you are going to culture under the laboratory condition, then with a suitable medium, it is not, what is that? It is not suitable for all the mediums. A particular medium, it will be essential for or used for the anther culture. So that is very, very important. Under the laboratory condition, that culture is called as what is that? Anther culture. Okay. This technique was first used in India. This is the NEET question, my dear students. To produce the haploid of Datura inaxia. The plant name is called as Datura inaxia by Guha and also Mageshwari. Here the name is a female name, but he is a male scientist. This is also one of the question in NEET. Mageshwari is the male or female? Mageshwari is the male scientist. Which plant they have used? In Datura inaxia. Okay, Datura inaxia. That is very, very important, my dear students. Datura family already studied, isn't it? Datura methyl. Tamil we may call as Umattai. Okay. The flower is also already known, but the structure of like this Datura plant is look like this one. It has a, what is that? Gamma petals, united in nature. Okay, united in nature. The seeds also, you know about the capsule nature. Seeds also in a capsule like structure and also have a, what is that? There is a thorn like structure. Okay, there is a small. Okay. So this is the Datura species. Already you know about one of the most important opioid. Okay, Datura species also there. See, so the haploids are completely sterile and no direct value. So that's why, suppose if it is present in single, okay, haploid only the male gamete only present in it. Take an example of the dioecious plants. The male plant is also separate and female is also separate manner. What happened? There is no use. That's why it is also sterile. When it come in contact with us, that means what does that mean? Both the gametes united together. Both the gametes joined together, then only the fuse together. So zygote formation takes place, isn't it? So it is useful one. So that as given here, the haploid are completely sterile and no direct value. The haploid are important as they are used to produce a homozygous line. Already we discussed homozygous means only N type of. Suppose when you are doing this one in the laboratory condition, when you are going to culture this anther in the laboratory conditions, what happened? No, only the anther only produces, isn't it, my dear? The ovary it will not produce. What is that meaning? That is nothing but the homozygous. Only one type of gametes will be produced. One type of gametes, isn't it, my dear? So this strategy can be easily integrated into breeding programs. Breeding programs. So this is the first step. You should note it down here. There is a three things is very, very important, my dear student. Anther culture, particularly it is a haploid nature, first of all. They need a suitable medium, second point. Which plant they have taken? Datura inaxia. Who is the scientist? That means Indian scientist means Guha and also Mageshwari. Third point. Mageshwari is, what does that mean? Male scientist. Which year? 1964. Completely will get the sterile conditions. Okay. So when you are going to culture the anther culture, you will get it, which one, homozygous line only. What is the meaning of homozygous? Only one type of gametes only, we will get it. That is very, very important. Okay, my dear, go to the next one. What is the main advantage of the micropropagation? So that's all about the culture, types of culture. What are the types of culture? Somatic culture, anther culture, and also protoplasmic fission. All those things already we discussed, isn't it? What is the main advantages? What is the beneficial effect of the micropropagation? Micropropagation. Propagation is nothing but the proliferation. Micro means very minute level, minute cells also. Take an example, a single cell into plant, entire plant. This is called as micropropagation. See here, the micropropagation is widely used in forestry and floriculture, flower. Okay. Okay, flower and forest. The micropropagations can also be used to conserve a rare or endangered plant species. Already we discussed this part also. Conserve. Okay, for future. Okay, future. And maintain the ecosystem. That is also very, very important. And some of the endangered species also they can conserve in the conserved by the micropropagation. It is also one of the important points. And third point is large scale growth of the plant cell in liquid culture inside bioreactor as a source of secondary metabolism. Already we discussed this point. Okay. Micropropagations can also be used to cross widely related species by protoplast fission. So they are called as what is a cybrid. What is the meaning of cybrid? Is cytoplasm plus hybridization. Two cytoplasm. Take an example, whether it may be a related species. That's why I told you pomato. Okay, my dear. Pomato is one of the plant that is potato as well as the tomato. So these two are combined together. You will get the cybrid. This is also one of the cybrid culture. See the next one. So this is also, these things also already we discussed here. The dianthus, chrysanthemum, gladiolus, and sweet potato, and diaceros, and manicid excellent. These are the things. Somatic embryogenesis. That things also already we discussed, isn't it, my dear? Next one is, we will pass on to the next one is, what is it, somatic hybridization. Protoplasm culture and somatic hybridization. Somatic means, already I told you, a body cell. Okay, somatic cell otherwise called as body cells. Only the mitotic cell division takes place. Isn't it, mate? See here, the protoplasm, also known as the naked plant cell, refer to all the components of the plant cell excluding the cell wall. The protoplasm is nothing but the naked plant cell. Just to take an example, any one of the plant cells, you can take an example. The outside there is a cell wall is there. The cell wall is made up of what is that, cellulose, isn't it, my dear? But inside what happened, there is a nucleus is there. There is a outermost covering is also there. What is the mean by protoplast? The protoplast is also known as naked plant cell. Naked plant cell means, what does that mean? It is nothing but the simple cell like this without cell wall. Okay. So what happened inside? What are the plant cell? It has accessory organelles, cell bound organelles, cytoplasmic organelle present in it. All those things present inside. This is called as protoplasm, but they are having the capability. See, excluding the cell wall. That is very, very important. Protoplasm is the naked plant cell, excluding the cell wall. These are the 2 points is very, very important. A hybrid procedure for fusion of somatic cell of two varieties of species is called a somatic hybridization. What is my somatic hybridization? Yesterday we saw it, isn't it? Pomato. Already saw it. Two plants. Take an example, A sample as well as the B sample. Collect the plant cell. A plant and B plant. After that, you have to isolate the protoplasm of A, protoplasm of B. fuse together with the help of PEG. Already told you, it is one of the gelling agent, polyethylene glycol. Without this one, this is the fusion agent, fusing agent, polyethylene glycol. So what happened? Two genes are united together. Two genes are united and introduced into the, which one the one of the host cell. Already we discussed in biotechnology, isn't it? Host cell. Particularly, the E. coli is the very good examples of the host cell. We introduce inside and go the multiplication. You will get both the characteristics in single plant. Okay. So that is called a somatic hybridizations. The first step in somatic hybridization is the removal of the cell wall by digestion with the combination of pectinase and cellulase. Here I told you, isn't it? The plant cell wall is made up of, just imagine, okay, for example, here take an example, plant A, okay, plant B. Okay, take an examples, isn't it, my dear? So this is a plant leaves. This one is also plant leaves. A plant cell. Yeah. Selecting. Okay, inside what are the things is there? Report. The outermost cell is made up of which one? Cellulose, because plant cells are made up of cellulose is the one of the polysaccharide. It is also having structural polysaccharide, similar to that of the plant B also, because it is also plant, it is also made up of which one? Cellulose. The structure will be like this. Same. Okay. Inside there is a nucleus is the, that's things. Okay, my dear. Okay, this is also made up of cellulose because it is also a plant. It is okay. I have to isolate the naked one. Naked one means just only we are taking the protoplasm. How it will be taken after the digestion of the cellulose, isn't it? How it will be digested with the help of the enzyme is called a cellulase. When you are introducing the cellulase, what happens? The outermost layer is completely eroded. You will get it the naked protoplasm. Isn't it? Likewise, the other plants also, other hands also, you will get it. This is the protoplasm. So this is the A protoplast. This is called as, what is that? B. Protoplast samples. Okay. So it has the parental characteristics. That is very, very important. It has the property, the parental, what they are having. So same things is there. These two are, what is that? Identified and also fused together with the help of the PEG. Already I told you, polyethylene glycol. So what is the meaning of this one? Polyethylene glycol. COL. Glycol. Simply called as PEG. It's a fusing agent. Okay. After the fusion, what happened? You will get it. How it will be? Look like this one. Combination of both. Okay. Combination of both means how it will be like this. This is the A sample. So this is the B sample. A plasmid, protoplasm, and B protoplasm. This side is B. This side is a, just imagine A. So it can be introduced into the host cell. It is nothing but the host. What is that? E. coli. With the help of the, but there are gene con and so on. So already but that things also, isn't it, my dear? Gene con and so on. So inside what happened? There are so many copies, multiplication takes place. So many copies of the combined form of the gene, isn't it? It's produced inside. Okay. Finally, it introduced into the plant. Finally, it introduced into the plant. How it will be look like this one? The plant look like This how it will be. The upper side, there is a plant, and lower side is also, you have the different shapes. Lower side there is a potato, and upper side there is a tomato. So already we discussed this part. So we'll get these two characters already individually present, isn't it, my dear? Now you will get the same plant, and you will get it these two plant characteristics. That is very, very important. That things has given here, my dear. Okay. Go to the next one. See this one. Systematic hybridization. Cellulase, the plants. So already in the cell lacking cell wall are called as protoplasts. That's why I told you. So without cell wall, what happened now? It is considered as a protoplast. See, these are the different types of plant material they have taken. Protoplast isolation, protoplast transcription, protoplast incubation. See, these are the plants. See the different types of plant is there. It has 4 types of plant is there. Here, these are the 2 plants. These are the 2 plants. We are placing it. Stripes, vacuum filtration, enzymatic digestion. Enzymatic digestion, what we are using here? Cellulase. After that, what happened? It will be transferred to the nylon mesh filtration. Then checking. Then transfections. Protoplasmics DNA, so the combination of the 2, A and B. Then solution, stop transfections. Wall culture. Again, incubation. So protoplasmic application process here, the PEG, all those things. These are the steps they were followed in the laboratory conditions, my dear. When you are going to study in the deep manner, this much no need, but is it note it down? Here you must note it down. 2 things is there. What is that? Which enzyme digestion takes place? Cellulose. Okay, cellulase, because it's act upon the only substance. The plant cell wall is made up of cellulose. Okay, my dear. Then which one is that? Fusing agent. PEG, polyethylene glycol. Okay, host, E. coli. Okay, so how many temperatures? Sometimes they may ask the question, 25 degrees Celsius up to 2 to 48 hours. Okay, my dear. See this. And protoplasmic fusion, see here, it involves the mixing of protoplast. Okay, mixing of protoplast, 2 different genomes. I already told you, 2 different genomes, isn't it? So 2 different genomes. And can be achieved by the either spontaneous or induced fusion method. Spontaneous, sometimes it happens in naturally. Otherwise, manually. Suppose when you are doing in the lab conditions, it is nothing but the manual aspects only. How it will be induced? What are the methods? See here. First of all, induced fusion method. The treatment with sodium nitrate. They are using different components, sodium nitrate. Calcium ion at high pH. Already told you. The polyethylene glycol, PEG. Electrofusion. What is the meaning of electrofusion? A sample as well as the B sample, how it will be fused with the help of, what is that? Micro syringe. Micro syringe. Or what is that? Micro pipette or gene gun. What is that? Gene gun. Okay, gene gun. Okay, gene gun. Okay, my dear. See, these are the important components. So, what are the main significance of the protoplasmic fusion? See here, when the fused protoplasm or culture on the suitable medium, see that after the fusion, you have to introduce in the medium. They regenerate cell wall and begin to divide to ultimately produce plantlets. So this is very, why are you introducing in the medium, So already you know a little bit the protoplasm, it contains lack of cell wall, isn't it? When you are introducing the medium, what happens? Automatically the cell wall components also, what happened? Automatically regenerated. After the regenerated only, what happened? The plant cell undergo the cell division process. Okay, that is very, very important. The somatic hybridization allow the productions of hybrids between the lines and species that cannot be produced normally by means of sexual hybridization. For example, the somatic hybrid between the non-flowering clone of potato and the flowering of potato clone produce fertile flowers. Similarly, somatic hybrid were produced between the rice and carrot. See the combination, rice and carrot. In both these cases, sexual hybrids cannot be produced. Somatic hybrids may be used for the gene transfer, transfer of cytoplasm, and the production of, which one you know, the useful allopolyploid. What is the mean by allo? Different species. The gene transfer takes place during the hybridization process. That process is called as alloploidy, allopolyploidy. This is very, very important, my dear. Somatic hybrids may be used to gene transfer. So somatic hybridization is also one of the allopolyploidy. Don't forget, my dear student, what are the plants they have taken for their experiment methodology. That thing's also very, very important. See here. This is the cell. This is the A plant. Okay, my dear. A plant. And also, this one is a B. Sample A. This one is called as B. We are selecting the cells. We are isolating the cell from A plant as well as the B plant. They also undergo the, which one you know, isolation of the protoplasm. Before that, you have to use the cellulose. Cellulase enzyme. They only act upon the plant cell, isn't it? The plant cell. So that time what happened, you will get it the protoplast. After that, what have to do? What we are going to do? We are selecting the different samples. This is a B sample. This one is a sample. Both are undergo the fusion process. So that is also PEG. Already told you, PEG. After that, undergo, which one you know? Cell division process. Okay. After that, it is also another process, cell division process, and also automatically stimulate the divisions and the differentiation of the hybrids. This process. Then again, it will be hybrids of leaves. It is also here the cell to the entire plant. Okay, major. This is very, very important medicine. This is also formed from the somatic cell hybrid. Somatic cell means body cells. Here, only we are taking the body cell of A, body cell of B plant. It may be fused together. The nucleus is also fused together. Okay, with the help of the specific chemical substances. Okay. And also, what are the previous process also, the forcing process and hygienic process also already we have to do before the experiment we start. After that, you will get it the plant. Like this is only hybrid leaves, similar to that of the parent. Okay, so it is very, very important, my dear. See that same process. See this one, the cell wall, vacuole, all this plant cell A, plant cell B is also there. We have to remove the, which one you know, outermost layer, that is cellulose and pectinase is a activate upon it. As a result, what have completely removed of the cell wall, you will get it the naked protoplasm. After that, parent A, parent B, you will get it two parents. So united to the PEG, PEC. Then the cell fusion, nuclear fusion, and somatic hybrid cells also undergo the medium. Then cell division is also that outermost cell wall is again regenerated. So the cell division started. Finally, we'll get it. Which one you know? Young plantlets. Okay, media. This is very, very important. Don't forget. Next, we are going to discuss about the synthetic seeds also. It is another important process, I think, my dear student. Synthetic seed. The somatic embryo are used to produce artificial. So somatic embryo also used to produce an artificial seed, just like a normal seed-like structure. Okay. So sodium alginate and calcium chloride are used to produce a seed coat of the somatic embryo. For example, so this is considered somatic embryo means, what is the meaning of somatic embryo? We are taking the plant cell, isn't it, my dear? The plant cell, there is a seed habitat also we produce inside the lab. This is also and treated with the two components. What is that? You know, sodium alginate. Note it down, my students. This is very, very important. Sodium alginate as well as the calcium chloride, as well as calcium chloride, or produce a seed coat. Again, what happened? It's also produce a seed coat during these favorable conditions. This is considered as a seed coat. Produced, that is very, very important. Produced under the treatment of calcium chloride and sodium alginate. So these are the capable of producing plantlets from the soil bed, just like the natural seeds. After that, what happened? It has the capability to produce the plantlets. So these seeds, they can produce the, what is that? Plants. Let's, just like a normal seed. Okay, so this is also an important idea. Biotransformation is also another method. So here it is ability to plant cell culture to catalyze the transformation of the readily available or inexpensive low-cost precursor into valuable high-cost final product, such as secondary metabolites, alkaloids, terpenoids, steroids, anthocyanins, glycosides, and polyphenols, etc. Examples. So suppose if we want to produce different components, means these are all are mostly found in which one, the colored substances, colored. Okay, for example, pumpkin. Okay, pumpkin. And also take an example, carrot. Which one they are having colors, orange, red, violet, yellow, munch, and also purples. All those things, they are all comes under the anthocyanin, glycos, and also terpenoids, alkaloids. There are so many alkaloids have been identified. Take an example, curcumin is also one of the alkaloids mostly present in the curcuma longa plant. Terpenoid is also so many terpenoids present in it, steroid component. Anthocyanin, blue-green. Glycosides, already some of the poisonous substances also present in it due to the glycosides look like a blue-green algae, blue-green like success, the poisons like success. Polyphenols, examples. Okay, these are the things. See here, the cell culture of stevia reboundina. Stevia reboundina can convert steviol into stevioside, alcohol into aldehyde form, which is 100 times sweeter than canned sugar. Take an example. This is the one of the plant. Very, very important. This is one of the plant. It contains the, which one, the conversion of steviol into stevioside. Steviol into stevioside. When it converted into steviol into steviosides, which is 100 times sweeter than the canned sugar. Okay. Just like a saccharin. And another one is plant tissue culture have been used to achieving a specific biotransformation. Biotransformation means, what is the meaning? The sugar habitat, the sugar content, sugar characteristics, sugar properties is also transfer from one plant to another plant. That is also biotransformation. Bio means gene transformation. Particularly, which one in there? Which gene is responsible for the sweetness? In the production of the cardiovascular steroid, digoxin, see the steroid, digoxin from the digitoxin, from digitoxin obtained from the digitalis lanata. Okay, digitoxin lanata. This is also one of the NEET idea. The digitoxin is also isolated from which plant? And another one is crozin. Take an example, crozin and picrocrozin from the culture of Crocus sativa. This is one of the plants, sativa. Atrobin palladona from the, see that, atrobin from the culture of Atroba palladona. And vinplastin is one of the cancer drug, anti-cancer drugs. Ajmalathin, otherwise called as jamalthin. Ajmalacin from the culture of Chrysanthus rosas. Chrysanthus rosas. These are the bioinformation. Bioinformation, what is a living organisms utilize these components for their health. Okay, it can be produced in the laboratory aspect with the help of the hybridization process. That's why it is called as biotransformation. Plant to animal, animal to plant, plant to plant. Okay, or same species or different species. That is very, very important. This is very, very important, my dear students. NEET point of view. Sometimes they ask the questions. Okay, so don't take it. The Atrobapelladon. Just imagine, this is also one of the important things. Already you studied in the when you're studying the families, types, families, Solanaceae, Fabaceae, Malvaceae, okay, Monocotyledon, listen it, my dears. Crocus sativa, it is also very, very important. Economically important. These are all are comes under the economical important. Okay, economical important. Okay, my dears, this is very, very important. Okay, see here. Another. So important components also given here, my dear. So Toxol, the anti-cancer drugs. Sometimes they ask the question, which is considered here? NEET. This is also one of the NEET. And one more is that, take an example, so apple. Okay, apple. Okay, take an example, apple. Just you made a cut. What is that? Just you made a cut. What happened now? You will get the structure, isn't it? So left for a few minutes, just leave it for a few minutes. What will happen after? Sometimes what happen? The outermost layer is covered by a dark brown in color. Isn't it? Dark brown color, isn't it? After a few minutes, just left it for, after cutting, just left for a few minutes. What happened? The layer is formation takes place, brown in color. That condition is called as what is a tamoxifen. It is one of the anti-cancer drugs. Okay, likewise, Toxol. That's why the doctor said, isn't it? Daily you take a apple. What happened? No, doctors no need, isn't it, my dear? Likewise, cigar, spiconines. It's one of the red dyes from the culture of Listospermum root. Okay, Listospermum root. And scopolamine from the culture of Datura quinine. Okay, from the culture of cinchona. Cinchona is one of the drugs used for malaria. Isn't it, my dear? This is very, very important. Pyrethrin. So this is also another one. Pyrethrin. These two are very, very important. From the culture of Chrysanthemum cinerea folium. This is the entire plant name. See here. Chrysanthemum cinerea folium. Okay, these are the informations. These are the important points, important components also obtained from the different plants, alkaloids. Okay, cinchona. Tetraquinine. Quinine is one of the alkaloid components. Okay, it is used for the cinchona plant. It is a plant. Cinchona plant, we will obtain. Quinine is a substance. Okay, they used as a drugs for malaria. And next important thing, cybrid, already we discussed. What is the meaning of cybrid? Cybrid is nothing but the cytoplasm hybrid is called as cybrid, just like a protoplasm. The same things as given here. Last time we discussed what is by cybrid. Okay, so protoplasm. So the somatic hybrids, the cytoplasm is derived from the both the parent. Same process. Cytoplasm hybrid is called as cybrid. Okay. However, the somatic hybrid can be obtained where nucleus is derived from one parent and the cytoplasm is derived from the both. See, this one is called a cytoplasm of both A cell, B cell. Thus produces cytoplasm. When united together, we will get this one. This is called as hetero. This one is homo. This is also homo, isn't it? It's in single. So both are united means you will get the heterokaryons. What is by karyon? Nucleus division. This is considered a nucleus. This is a nucleus. Center there, the cell it contains. What is that? Nucleus, both. Isn't it, my dear? Both the nucleus united together. That is called as heterokaryon. Heterokaryon means both are united. A plus B united here. Then what is man? Synkaryocyte. Heteroplast. These are the two types. Heteroplast means you will get it. What is that? A plus B, both are united together. So this is the part of A, this is the part of B. A part nucleus and B part nucleus. That's why it is called as synkaryocyte, hybrid nature. This is cybrid. What is the meaning of cybrid? Only cytoplasm only mixed here. There is no nucleus mixation takes place in this part. So this is the different types. Here nucleus is fused. A and B cells are fused. That is the, what is the reason of this one? A and B also united. That is also for, what is that? The nucleus also united here. That's why it is called as synkaryotype. Karyon means nucleus. Isn't it, my dear? But here, what heteroplast only? What is the meaning of heteroplast here? There is no combination of, there is no unison of, what is that? Nucleus. Nucleus, it's not united here, not united. Instead of nucleus, what happened, the cytoplasm only united here. That is called a cybrid. Two cytoplasm of the different species united to form a heteroplast. That condition is called as cybrid. Okay, two cytoplasm only united. Nucleus, it's not united. Okay, my dear, don't forget. This is very, very important. Same process here. A cytoplasm hybrid, that is called a cybrid. Cytoplasm hybrid is simply called a cybrid. A and B fusion. So here, what happened? This one is called as ketosis. You will get it this type of plant. See this? These are all are called as, what is that? Cybrid. Okay, cybrid variety. We will obtain from the same species. Okay, media. So this is the example. And next one is the somoclonal variations. Somoclonal variation. How it will be vary? The variability generated by the use of tissue culture cycle has been termed somoclonal variations. Suppose we will not get it, we will not get a same things. We should generate by use of tissue culture. We will get it the different types of variability we need. Means what you have to do? We can choose the tissue culture cycle. Has been termed somoclonal variations. All plants arising from the tissue culture should be exact copy of the parental plant. Isn't it, my dear? This is the exactly happen. What happened in the lab as well as naturally. However, phenotypic variant were frequently observed among the regenerated plants. Sometimes what happened, phenotype means physical variations also we will obtain during the time of regenerated plants, regenerated. The causes of variation is attributed to change in the chromosome number and structure. This is very, very important. How it will be changed? The regeneration plants also, what happened? How it will be vary? Variations occurs only the gene level, then only gene level. Then only the chromosome number structures variation takes place, means it also expressed in the phenotypical aspects. And implications of somoclonal variation is breeding, so that novel variant can arise, then this can be agronomically used. So this variation is also used by the agronomically. Agronomically means agriculturally. Okay, Media. Sometimes such variation lead to the disease resistance and herbicide resistance and also insect resistance. This is the important. What is the somoclonal variation? How it will be utilized? What is the main aim of the variations? Sometimes such variation, which variation? This gene level variations lead to the disease resistance, herbicide resistance, and also another one is called as insect resistance. This is the ultimate aim. Okay, my dear, this is very, very important. Okay, so what is the next one? See, already we discussed. So crop and species, there are so many gene bank also is there. Sometimes they ask the question from this point. Don't forget, my dear, this is very, very important. First of all, is noted on wheat, where there is a gene location bank is center is present in a DWR. Kernel, it is also Haryana. Rice, already we know about Central Rice Research Institute, Cuttack, Orissa, very, very important because it's rice. Potato, okay, see here. CPRI, Central Potato Research Institute, Simla, because it's a hills. Potato only they grow in the hills place only. Himachal Pradesh. This is also very, very important. This is asked so many times. Cuttack. And also Philippines also, I will tell you. Cotton. Cotton Research. Central Institute of Cotton Research. Nagpur, Maharashtra. Take an example. Maharashtra. Okay, Mumbai. Which is the ancestor of the India, the Maharashtra, because presence of cotton industries. That is very, very important. Sugar cane, very, very important, my dear students, in Tamil Nadu. Okay. The SBI. Okay. Coimbatore, Tamil Nadu. Tobacco, Central Tobacco Research Institute, Rajamundry, Andhra Pradesh. Maximum tobacco is Andhra Pradesh only, AP. And also chili. Okay. And pulses, see here, the Kanpur, Uttar Pradesh, UP. And forage crabs, see here, Jhansi. Plantation crabs, see that, Kasargod, Kerala. Plantation, plant only, forest, imagine. Tuber crabs, see that, Trivandrum. Tuber. Oil seed crabs, very, very important, oil seeds, Hyderabad. Horticultural crabs, okay, Indian Institute of Horticulture, Bangalore, Karnataka, don't forget. And sorghum is one of the wheat, isn't it, my NRC, sorghum, Hyderabad, Andhra Pradesh. Groundnut, see that, Gujarat. And soya beans, so Indore, Madhya Pradesh. Maize, it is also very, very important. Indian Agriculture Research Institute. See here, rice is also very, very important. So here asked the Indian Agriculture Research Institutes, where it located. So many times they were asked the question, NEET. Oil seed also. Oil seed. It is also Andhra Pradesh. This is also NEET. Particularly two things is very, very important, my dear students. First of all, rice is very, very important. And cotton is also sugar cane, oil seed, and soya beans and maize. Okay, just to go through it, you will get it the idea. Okay, these are all just you practice it more than 3 times. Just read it. Okay, best foodstuffs, crabs, rice and wheat and potato, maize. Okay, these are the very, very important. Okay, so these are the, already we studied these things. See, Central Potato Research in Simla, Central Plantation and Craft Research in Kasargat, Central Institute for Cotton Research Nagpur, Tobacco Research Rajamundry, Indian Agriculture Research in New Delhi, Indian Grassland Forest Research in Jhansi. Okay, these things also, National Bureau of Plant Genetic Research and the NPPGR establishment in the year 1976. These are all not clear. Just to go through your book, you will get the idea. Next, we will pass on to the next one is very, very important, my dear student, germplasms. What is the main importance of germplasm? It is also similar to that of the which one, you know, hereditary of the one plant to another. Germplasm. See here, the sum total of the hereditary material, that is called all the alleles of various genes present in the crop species, and its wild relatively it's referred to as germplasm. Which one, you know? All the alleles, various genes present in the crop species. Okay, how it will be vary, how it will be relative. That is called as, what is that? That study is called as germplasm. The germplasm consists of land races, modern cultivars, obsolete cultivars, breeding stocks, wild forms, and wild species of cultivated crops. Where it will be used? These are the field. The germplasm include both cultivated and wild species and relative of crop plants. Germplasm is collected from the center of diversity, gene bank, gene sanctuaries, farmers field, marker, and also seed companies. What is the main? How it will be collected? Germs. Germs is nothing but the seed. Okay. It may be whether plant or whatever it may be. Germplasm is the basic material for the launching of crop improvement program. Germ is the basic material. Which one? Without seed, without sperm, without egg, we will not produce the new variety. Okay. That is very, very. Germplasm may be indigenous, collected within country. Indigenous means within the country. Exotic species, already we discussed, collect from the foreign countries. These are the germplasm is also two types. What are they? Indigenous, within the country, collected. Which one, you know? Seeds or germs or sperm or egg. It is also called inbreeding, like that. Inbreeding means same species within the country. Okay. Exotic species means outside from. Okay. The genetic pool represents the entire genetic variability or the diversity availability. Okay. Availability. Also very, very important. These are all are called as what is that? Germplasm, my dear. This is very, very important, my dear student. Germplasm must be a question. Yeah, single question. Germplasm conservation. How it will be germplasm conservation is also followed. See here, the conservation refers to the protection of genetic diversity. Suppose the plant, they are having the different diversity, isn't it? Whether it may be transferred through the commercial or whatever it may be, onion, orange, or whatever it may be. Coffee Arabica. Coffee Arabica. What is the meaning of coffee Arabica? It is comes from where, you know, Arabian. Okay. Take an example of this. Pisum sativum, isn't it? So indica, like that, isn't it? Asatictica indica. What is that? Indica means what is that? It's derived from it. It is also discovered from India. Suppose the cross between these two, the Arabic country, the seeds also, that coffee seeds also comes from where, you know, Arabia into where, you know, in Indian countries also we are cultivating, isn't it? It comes from the where, you know, when, you know, once upon a time, the ancestor times, that Jahangir. In historical aspect, just go through it, you will get the ideas. Coffee Arabia is also arrived from where, you know, Arabia, in which period, you know, Jahangir period, like that, it may be mentioned. The conservation refers to the production of genetic diversity of the crop plant from genetic erosion. For example, some of the plants completely, what is that? Disappear, exotic species. It is also regenerated. There are two important methods of germplasm conservation or preservation. Two types already we discussed, in situ and ex situ. What is the meaning of in situ? Conservation of germplasm under natural condition is referred to as in situ conservation. Natural conditions. Ex situ, it is referred to the preservation of germplasm in gene bank. This is the most practical method of germplasm conservation. This is the cheap method of the germplasm conservation. Sometimes they may ask the question, which one of the following method mostly in very cheap method of germplasm conservation? Like that, they may ask the question ex situ. What is the meaning of ex situ? Ex situ is nothing but the germplasm is also conserved in the gene bank. Already we studied in the biotechnology, genetic bank, gene library. Like that things also already we discussed, isn't it, my dear? See, this is considered germplasm conservation. These are the different types of number has given, isn't it? These are all are considered as a capsicum. See here, 13, 36, 13, 37, 38, 39, 40, 41. All are same species. These are all also ones beans types. Okay, here take an example, tomato, pineapples, and also the onion. Germplasm conservation means we have to conserve the seed or germs in two different parts, ex situ, in situ. See that seed bank. This is considered a seed bank. Each one it has a specific storage ability, selectively, highly selectively. That is superficial characteristic seed only is taken and stored for what is that? Stored in seed bank. Once what happened, natural disaster happened, completely, the world is completely eroded or destroyed, means we can use, suppose they may grow, the seeds or grow for new world, for the formation of the new world. See, this is considered a gene bank. Okay, so inside the gene bank look like this one. Okay, completely preserved. That is very, very, very important, my dear students. See here, gene bank. The germplasm is stored as a seed of various genotypes. Different types of genotype. What is the meaning of genotype? Gene means genetically different. That is very, very important. Take an examples. IR 8, IR 34. Like that. What is the variation of this one? Depends upon the gene only. It can be grow. What is that? Tall, dwarf, semi-dwarf. Already we discussed, isn't it? So these are the three things. Is that the tall is maximum 15 feet. Just imagine. This one is called a 7 feet. This one is called a 3.5 feet. So likewise, the seed bank is germplasm is stored at a seed of various genotype. Plant bank. What is the meaning of plant bank? Plant is also how? It is a field in which the fruit trees or crabs are grown. Shoot tip banks. Germplasm is considered as a slow growth culture of shoot tips and node segments. This is also another part. Cell and organ banks. A germplasm collection based on their cryopreservation. What is the meaning of cryopreservation? Cryopreservation is nothing but the storage of germs under low temperature. Which one you know? Minus 196 degrees Celsius. Liquid. Already we study liquid. Nitrogen. Don't forget, my dear. So many times they ask the question, 4 or 5 times. NEET question this one. DNA bank already in our book. Already we studied in the where? Biotechnology, gene technology, isn't it? So in this bank, DNA segment from the genome are maintained and conserved. Okay, these are the important, my dear students. Don't forget. What are the bank is that? Seed bank, plant bank, shoot tip banks, cell and organ banks, and DNA banks. Okay. So what are the some international institute conserving germplasm? What are the institute present in? Where it present in? Sometimes they may ask the question in this part. International Rice Research Institute, already you know about. International, what is the Indian? Okay, IRRI, Las Banas, Philippines, very, very important, my dear students. The world level rice research institute, that is called as International Rice Research Institute, simply called as IRRI, Las Banas, Philippines. The Central International Measurement, Demise, Trigo. So that is the expansion of CIMMYT. Central International Demeasurement, Demise, Trigo. El Baton, Mexico. The double M is considered Mexico. Okay. And then International Plant Genetic Research Institute, Rome, Italy. This is also important, my dear. These three things. Expandable question, expand aspects, they ask the questions. International Rice Institute must. Philippines, don't forget. Okay. India, Cuttack, Orissa, don't forget. Okay, my dears. See, this is the plant bank. How it will be look like? The plant bank. What happened now? See that plant name. Different types of plants also stored in a lab, in culture. See, the lower side, there is a agar medium. Each one it has a specific medium. This one is called as agar medium. Okay. Okay, this one is plantlets. The single bottle, it contains how many plantlets? Just you can imagine, at least you can calculate up to 25 plantlets. We may just 25, I told. In this plant, let's say what happened when you are going to hardening. Take an example. How many plants here? Just to take an example, this 4 bottle, you will get 100 plantlets. Isn't it? Take an example, total overall. It may be 1 million or 10 million. Okay, it can put like this. How many plantlets is that? Within the short, what is that? Period or time or area, like that, very, very important. See, this is the considered DNA bank. Okay, this is also a DNA bank only for plantlets here, only plant. Some of the animals plant samples also collected. These are the DNA samples. Okay, this is for animals. Okay, see that germplasm conservation approaches. There are 2 types of germplasm conservation. Is there? One is called as ex situ, another one is called as in situ. Ex situ conservation is called as outside. In situ is called as inside. Okay, this is natural habitat. This is for the away from the natural habitat. The ex situ divided into 3. One is called as conventional, in vitro, and field gene banks. The conventional orthodox seeds, ancient. The in vitro recalcitrant seeds, cloned DNA, pollen. These are the things. In vitro means lab. Field gene means plant conservation. Suppose the seeds also planted and also conserve as a forest or a botanical garden, like that. See here, they have given as a botanical garden or arboriculture, arboriculture. And also recalcitrant seeds also again divided into 3. The tissue culture methodology, cryopreservation, and slow growth. These are the 3 methods they were used. These are all are considered notes ex situ. The in situ, that is also 2 things is there. One is called a natural habitat, another one is called farming. Natural means farmer field or home garden. Sorry, natural habitat means it's a wildlife sanctuaries, natural parks, and biosphere reserve, genetic reserves. These are all naturally present. Wildlife sanctuaries, Mudumalai, Periyar. Okay, Carpet, Simla, Kanham. So many national parks is also there. National wildlife sanctuaries is also present. Natural parks is also. Biosphere reserves, genetic reserves. These are all our natural habitats. On farm means farmer field, just like our field, agriculture field. Home garden, we are maintaining our home garden, isn't it? Tribal area. These are the important things, my dear students. Okay, so these are the germplasm conservation approaches. Next, we will pass on to the next one is called as, what is that? Question part, evaluation yourselves. Nowadays, plants grow by tissue culture are called as explant, somoclonal, and micropropagation, and androgenic haploid. Just imagine which one is there. Okay, see that? Somoclone, somatic clones. Nowadays, plant grow by tissue culture. Tissue culture is nothing but somatic nature. Explant, it will not come. Just imagine explant, it will not come. Okay, the explant, it will not come, my dear. Don't forget. And somatic clonal means, tissue culture already, somatic cell divided into gives entire plants. So this is a correct one. Micropropagation, it is also one of the micropropagation. It is not a micropropagation, tissue culture. And androgenic haploid, it is not an androgen, anther. Okay. And virus-free plant can be obtained through grafting. It is not possible. Callus culture, what is that? It is not possible or possible? It is possible. Shoe-tip culture, what is that? It will possible or not? See this one. Shoe-tip culture, possible. And third one is called a suspensor culture. Here, which one is the correct answer? These 3 are related to the normal. See, the grafting is not coming. Callus culture, sometimes it happens. What happened? No, virus. Suppose when you are doing shoe-tip culture is possible. Suspensor culture is not possible. Okay, virus-free plant can be obtained through only shoe-tip culture. So go to the next one. The somatic hybrid between potato and tomato, already you know, but this is a direct question, my dear students. Already we discussed, isn't it? Bomoto, mopoto, topomo. It's not come. So this one is the correct one. The plant pod used for culture is called as which one? Plant pot means it is a explant, not scion, not stalk, not callus. Callus is a mass of cells produced from the single cell. That is callus. What is my scion? What is my stalk? Okay, we will study these things. Which one it has a root? Suppose take an example, 2 plant is there. Which one it has a root? This is called a stalk. A plant. B plant is an alien species. It is comes from the another one. That is called a scion. Here the pot is this one only. The branch of pot you have to unite it here. This is the B plant. This one is called as A plant. A is called a stalk. Which one it has bearing the root? That is called a stalk. Which one is the branch only with leaf? That is called a scion. Okay. The pH of the nutrient medium already we discussed. Acid pH. Which one is the acid? All are acid only. But which one is related? Tissue culture means 5 to 5.8. Not low one, not near to the alkaline. So these two are what is that? Completely acid, very less in nature. Acid is high. This one is near to 6, 5 to 5.8, 5 to 5.8 only. This is the exact matching of the pH of the nutrient medium of the plant tissue culture. That is very, very important, my dear students. Okay, my dears, that's all I think, my dear students. We will finish it up to the level. Part 2 only finish it. We will pass on to the next one is part 3 in the next class, my dear. Okay. Today, what are the things already we discussed? You know very about. Once I repeat for you, just I will summarize all those things. So what are the things you know? First of all, Anther culture. So you know about who did that things also already in our Datura, Inaxia, Kuha, and also Mageshwari in the 1964. He is a male scientist. Okay. Advantages of micropropagations already in our about. Mostly used floriculture and forest culture. Endangered plant species also conserved. And secondary metabolism also we can prepare, and also cybrid propagation also done by the protoplasmic fission. That is also very, very important. Okay, some of the examples also given. Protoplasm culture also already called as somatic hybridations. How it will be taken already we discussed very deeply. So as a result, you will get it the tomato. Okay, and what are the inducer fusion methods? The treatment with the sodium nitrate before. After the calcium ion at the high pH, polyethylene glycol we used for the which purpose, you know, fusion agent. Okay, PEG. So finally we will get the plantlet, and also some of the examples also given. And allo polyploidy we will get it. That is called as different species united together form as a single species. You will get the different plant characteristics, properties in the single plant. Okay, already we saw it this part. Okay, very deeply. Which enzyme used for that? That is cellulose is an enzyme. PEG, that is very, very important. Okay, so already we discussed this part also. Synthetic seeds also. So with 2 components, it's a sodium alginate, sodium, and also calcium chloride. Very, very important for the seed coat formation. NEET question, my dear. Okay, biotransformations. Okay, toxols, cybrids. Okay, and also clonal variations. Already we discussed. What are the types of locations, gene bank centers? Already we discussed this part also. Okay, and germplasm is also germplasm conservation. There are 2 types of germplasm conservation: in situ and ex situ conservation. Okay, and seed bank. Okay, seed bank is just note it down. These are the very, very important: seed bank, plant bank, shoot tip bank, and cell and organ bank, and also DNA bank. Particularly note it down. This is minus 196 degrees Celsius liquid nitrogen. Very, very important. 3 things is very, very important. Some International Institute of Conservation of Germ Plants, International Rice Research Institute, IRRI, in where, you know, Las Banas and Philippines, Center International, D. Majorromenates, D. Maize, Trico, that is CIMMYT, L. Bodson, Mexico, International Plant Genetic Research Institute, Rome, Italy. These 2 things is very, very important. This one, they may ask the question in expandable manner. Plant bank, already we discussed how it will be stored. These are the complete, what is that? Mind map for the germplasm conservation approaches. That's all about, my dear students. Today, the part 2 is finished. We will see in the next class in part 3. Thank you so much. Bye. Take care.